Hello and good morning, everyone. I hope you are having a great day. I am Shane Mariel J. Caba, and together with my partner, Will and Geraldine Novecio, we're going to talk about archaeal viruses. For today's discussion, we're going to tackle these topics: the overview and history of archaeal viruses, the classification in its morphotypes, unique mechanism of variant release, evolution and evolutionary considerations, the importance of archaeal viruses, some immune system interactions, potential pathogenicity of archaeal viruses, and lastly, some major questions that are frequently asked about archaeal viruses. Before going over our key topics, let's define some terms first. So, di ba nga mong topic is archaeal virus? So, ato sa siyang bulagon. Bulagon na to siya into archaea and into virus. So, what is archaea? So, we all know that archaea, they are single-celled microorganisms with structure similar to bacteria, and they are famous for their love of living in extreme environments. Sa virus, sa previously nato na natapik ni Sir, it is an infectious agent that replicates only inside the living cells of an organism. So mag-replicate siya sa host, and it infects all life forms from animals, ranging from animals and plants to microorganisms, which includes the bacteria and the archaea. So archaea plus virus, we have archaeal virus. Ang archaeal virus is a virus that infects and replicates in archaea that are found in acidic hot springs. Kay love malagi sila mag-live sa extreme environments. So makita nato sila sa acidic hot springs, highly saline bodies of water, and we can also find them at the bottom of the ocean. So let's go over the introduction and a little bit of history about archaeal viruses. Archaea have been recognized as a third domain of life in addition to bacteria and eukaryotes 40 years ago. Although morphologically nearly indistinguishable from bacteria at the molecular level, archaea present a mixture of features, some of which are closely related to those of eukaryotes, and others are shared with bacteria, whereas some appear to be unique. Of the more than 6,284 viral isolates infecting prokaryotic hosts, only 117 infect archaea, and the vast majority of archaeal viruses have been isolated from members of Euarchaeota and Crenarchaeota. Many viruses replicating in Crenarchaeotes exhibit unusual variant morphologies, including the bottle and droplet shapes. Some are able to grow tails once released from their host. In contrast, over half of all characterized viruses replicating in Euarchaeotes resemble classical bacterial viruses with icosahedral heads and helical tails, and the remaining viruses belonging to this group have several unusual variant morphologies, suggesting Euarchaeal viruses may be more diverse than currently known. Archaea are now recognized as a major part of global ecosystems, contributing up to 20% of the total biomass on Earth, and are major players in global biogeochemical cycles like methanogenesis and ammonia oxidation. Most archaeal viruses have been isolated from two types of habitats: extreme geothermal environments and in hypersaline environments, where archaea tend to dominate over bacteria and eukaryotes. It has been suggested that archaea have evolved to survive in environments, both extreme and moderate, that impose high levels of chronic energy stress, which allows them to outcompete bacteria in such conditions. In summary, over the past 30 years, more than 100 archaeal viruses have been isolated. A brief history of archaeal viruses. The first description of an archaeal virus was made by Torsvik and Dundas in 1974 in a Nature paper titled Bacteriophage of Halobacterium salinarum. This virus, named HS1, is a codovirus identified as infecting Halobacterium salinarum, which is a halophilic archaeon. In 1980s, Wolfram Zillig and his colleagues began isolating viruses from thermophilic archaea of the orders Thermoporchalis and Sulfolobales. In total, he and his colleagues would discover and characterize four archaeal virus families, namely Fusiloviridae, Rhodiviridae, Lipothrix viridae, and Gutaviridae. Going to 1982, the SSV1 is the first fusilivirus described. Then in 2004, the Torivirus STIV1 was described. In 2005, the Bicodovirus ATV was described. In 2012, the Aeropyrum coil-shaped virus is the first spirovirus and the first known single-stranded DNA archaeal virus. In 2017, Sulfolobus alpha portoglobovirus is the first described portoglobovirus. And in 2020, the Adenivaria is the only virus realm to exclusively contain archaeal viruses.
And now let's proceed to the classification and morphotypes of archaeal viruses. One of the most prominent features of archaea is the extraordinary diversity of their DNA viruses. Many archaeal viruses differ substantially in morphology from bacterial and eukaryotic viruses and represent unique virus families. And all archaeal viruses isolated thus far have DNA genomes, so they are extremely diverse in terms of variant morphologies and gene contents. So these viruses are currently classified into 20 families, as shown here in this figure, whereas taxonomic classification of several additional groups remain to be assessed. So this figure is the representation of variant morphotypes of archaeal viruses infecting members of the phyla Euarchaeota and Crinarchaeota. The discovery of archaeal viruses has revealed a number of unique morphologies. So I will be discussing one by one their distinct morphotypes, and let's start first with the head-tail viruses. So similar to their bacterial relatives, variants of archaeal head-tailed viruses comprise an icosahedral capsid, which is the head, and a helical tail containing tail fibers, which play a role in cell recognition. So all head-tailed viruses encapsulate linear double-stranded DNA genomes, which in archaeal viruses can vary in length. So the archaeal head-tailed viruses have been isolated from extreme halophils as well as from mesophilic and marine hyperthermophilic methanogens. So far, only 5 viruses infecting methanogens have been isolated, and they all are either Mayo or Sipho viruses. So most haloarchaeal virus isolates also represent the head-tail type. So these viruses with all 3 prototypical tail morphologies have been isolated, such as long contractile tails typically of members of Meoviridae, short tails in the members of Podoviridae, and long non-contractile tails in Cephuviridae. So as shown here on these figures below. So next morphotypes is the tailless icosahedral viruses. Overall variant organization is the same for viruses from all 4 families of tailless archaeal viruses. The icosahedral protein capsid covers an internal membrane layer which encloses the double-stranded DNA genome. However, structural studies have revealed that the structures of the protein capsids are very different in viruses from different families. So based on this feature, the 4 families can be assigned to one of the 3 groups, which is the viruses with double jelly rule, which in the members of Toriviridae, viruses with 2 single jelly rule in Spherolipoviridae and Similoviridae, and viruses with a single jelly rule in Portogloboviridae. So I have here in figure A and B on the left side is the overall view of Sulfolobulus turreted icosahedral virus or STIV under family Toriviridae. So it reveals the internal structure of the STIV variants, which is T as turrets, P pentons, capsid, membrane, and genome. So in STIV, each penton base is further decorated by a turret-like appendage that protrudes above the capsid surface. So the turret complex participates in host recognition through binding to pili-like appendages on the host cell surface. And lastly, the figures in A and B to the right is the overall view of Archaeal hispanica icosahedral virus 2 or HHIV2 under family Sphaerolipoviridae. So the capsid is depicted in blue whereas the spike complex is in purple. So in figure B is the schematic cross-section of a Sphaerolipovirus particle revealing its internal organization. And now let's proceed to the linear viruses. So all characterized linear archaeal viruses infect hyperthermophilic Rhinarchaea. So these viruses can be divided into four groups such as filamentous, rod-shaped, coil-shaped, and bacilliform. So the filamentous and rod-shaped viruses are classified into the family Lipothrix Veridae and Rhodiviridae. So the variants of Aeropyrum pernix bacilliform virus 1 or APBV1 are stiff and bacilli-like, and the virus has been classified into a new viral family, Clavaviridae. Also, Aeropyrum coil-shaped virus or ACV, which is classified into the proposed families Paraviridae, has hollow cylinder-like variants formed by a coiling nucleoprotein fiber. So we have here the schematic representation of the SRIV2 particle under Rhodiviridae, revealing its internal structure, and the MCP or major capsid protein homodimers are depicted in green and red, whereas the linear double-stranded DNA is shown as a black line. The second figure is the schematic representation of the AFV1 virion, and the MCP heterodimers are depicted in purple and magenta. The membrane is shown in orange, and the linear double-stranded DNA is represented as a black line. Next morphotype is the spindle-shaped viruses. So they are widespread in archaea-dominated environments and represent a prominent component of the archaeal virus sphere. However, due to intrinsic flexibility and heterogeneity of the spindle-shaped virions, structural studies on these viruses remain highly challenging. So they are abundant in both hypersaline and hyperthermic environments, and these viruses have a short or long tail attached to one end of the virion or long tails attached to both ends. So for the family Fossiloviridae, it is the biggest group of spindle-shaped viruses and includes Crinarchaeal viruses with a short tail. And the members of the Fossiloviridae family have small lemon-shaped virions decorated with terminal fibers at one of the two pointed ends. So the type species is Sulfolobulus spindle-shaped virus 1 or SSV1, and the virion is rather flexible, although the presence of a lipid membrane is unclear. Also, the representatives of the Bicauda-Veridae family have tails of different lengths at one or both pointed ends of the lemon-shaped body. Remarkably, the prototypical virus of the family Acidianus two-tailed virus or ATV, the only member in the family Bicauda-Veridae, has an extraordinary capability to form the tails outside the host cells. So they undergoes a morphological transformation outside and independent of the host cell. The ATV variants are released from the host cell as tailless falciform particles, after which they develop extracellularly long tails at both ends of the spindle-shaped virion at temperature of the natural habitat. There are also unusual archaea-specific morphologies, so such as bottle and droplet-shaped viruses. So for example is the Acidianus bottle-shaped virus or ABV and Sulfolobulus newzealandicus droplet-shaped virus or SNDV infect hyperthermophilic Rhinarchaea and represent the viral families Ampulaviridae and Guttaviridae, respectively. So the bottle-shaped variant of ABV seems to be rather complex, and the genome is most likely in a nucleoprotein filament, forming a cone-like structure that is surrounded by an outer layer, and the tip of the bottle is formed by a separate structure. And lastly, this spherical viruses is represented by the viral family Globuluviridae, which includes Crinarchial pyrobaculum spherical virus or PSV and Thermoproteus tenax spherical virus 1 or TTSV1. So the virion consists of a lipid envelope surrounding a linear double-stranded DNA genome that forms the helical nucleoprotein complex. Thus, this architecture resembles that eukaryotic single-stranded RNA paramyxoviruses. And recently, a worldwide distributed group of pleomorphic viruses has been described. So these viruses has been designated pleolipoviruses based on their lipid envelope and pleomorphic appearance. So pleolipoviruses contain two or three major structural proteins and one of them forms spike-like structures on the virion surface, whereas the others are internal membrane proteins. So these viruses are non-lytic, only retarding the host growth, and they acquire their lipids unselectively from the host cell membrane. And consequently, it has been proposed that pleolipoviruses uses budding to exit infected cells. In the last decade, however, a major effort has been undertaken to study the archaeoviruses, and 42 viral species have now been described. So among these viruses, only one species possesses a single-stranded DNA genome, whereas the rest are double-stranded DNA viruses, so as shown here in this table. Thus far, RNA viruses that can replicate in the laboratory on archaeal hosts are unknown, despite the fact that a variety of RNA viruses infect bacteria and eukaryotes. So this table are the summary of the archaeal viruses that have been isolated and characterized thus far. And also this table is the continuation of the previous slide. It also shows the specific host of the archaeal viruses and their isolation sites. Nearly all known archaea-specific viruses infect hyperthermophiles of the phylum Crinarchaeota, and they have a range of unique morphologies that have never been observed among viruses that infect bacterial or eukaryotic cells, such as shapes that resemble bottles, spindles, droplets, and coils, in addition to morphologies common in the biosphere, such as spheres and filaments, so as shown here in these figures. The cosmopolitan viruses of the archaeal virusphere include head-tail viruses, viruses with icosahedral capsids and helical appendages attached to them, of the order Caudoviralis, and tailless icosahedral viruses of the families Sphaerolipoviridae and Turriviridae. So all of these viruses, except for turriviruses, infect members of the phylum Euarchaeota, and the biological basis for such host specificity remains unknown and difficult to understand, as some of the eukaryota inhabit extreme habitats similar to those of Crinarchaeota. And now let's proceed with their unique mechanism of virion release. One of the major contributions of archaeal virology has been the discovery of a new virion release mechanism from infected cells. Initially, archaeal viral release strategies were delineated based on host phylum, eukaryotic viruses, while crinarchaeal viruses were non-lytic, releasing from chronically infected cells by a poorly understood budding mechanisms that typically does not significantly affect host growth. And recently, the lysis pathway for two lytic crinarchaeal viruses has been described that included formation of remarkable lysis structures not previously seen in other viral systems. Moreover, the life cycles of these viruses have unusual features as revealed by the recent discovery of a novel virus egress mechanism that involves the formation of a specific pyramidal structures on the host cell surface. So during both STIV and SIRV2 infection, the seven-sided pyramid-like structures are produced that typically extend above the surface of the cell. So during lysis, the seven leaflets of the pyramid open like petals of flower, producing 100 nanometer holes through which newly formed virus particles exit the cell. So the production of the pyramid structures is solely dependent on a single viral protein, which is C92 in STIV and P98 in SRV2. As you can see here in the left figure, after the assembly of the virions they arrange into bundles containing the pyramidal structures form on the cell surface, and then perforating the S layer and disintegrate the virion bundles into separate mature virions, and then opening off the pyramidal structures, allowing virion egress. But recently, studies stated that it is unclear how pyramid formation disrupts the overlaying S layer, what controls the size of the pyramids, or the process that controls pyramid opening. So, most high temperature, low pH hot springs present numerous challenges that must be overcome by viruses and their host. As a result of being low nutrient environments in combination with high metal and or salt concentrations, most high temperature, low pH hot springs have cell densities lower than seawater. So this low host abundance makes it difficult for viruses to find a new host after release through lysis or budding from an infected cell. So the difficulty of finding a new host is further complicated by the fact that the half-life of archaeal viruses at high temperature is often short. So to overcome this challenge, some archaeal viruses have developed novel mechanisms to increase their ability to find host cells. And one example of this is the Acidianus two-tailed virus or ATV, which upon release from the host cell undergoes a dramatic conformational change where the central spindle shape of the virion contracts in width while the two tails extend in length up to 1 micrometer. So this morphological change occurs in the absence of the host cell, energy source, or external cofactors. Such a dramatic morphological change is hypothesized to assist the virus in its search for a new host by dramatically increasing the area it can search and by minimizing the time that the viral particle spends unprotected outside of a host cell.
Thank you for that, Will. Now let's go over the evolution and evolutionary considerations of archaeal viruses. Many unusual characteristics of archaeal viruses of hyperthermophiles likely represent adaptations required for replication in their host and for stability in extreme environmental conditions. Additionally, genetic mutations occur in archaeal viruses at a higher rate than bacterial DNA viruses and are more frequent in coding regions than non-coding regions, which may contribute to the phenotypic diversity of archaeal viruses. High temperature and low pH environments inhabited by archaea have a low density of cells. The half-life of archaeal viruses in high temperatures is often short, so some viruses have evolved mechanisms to overcome these challenges. Almost all cultured thermophilic archaeal viruses are capable of establishing chronic or persistent infection, but only a small portion are exclusively virulent. Halophilic archaeal viruses tend to be lytic but can also be lysogenic. This suggests that a lysogenic life cycle for archaeal viruses may be an adaptation to a harsh environment outside their hosts. Genomic sequences called virus and plasmid-related elements, or the VPRES, contain viruses and other MGEs like plasmids that integrate into the host genome, forming groups of genes that can move between viruses during recombination. This allows for new viruses to be created through reassortment, and this type of combination has been proposed for why haloarchaeal viruses within Halopanivirales and Pleiolepoviridae can have the same core structural proteins but different genomic characteristics and replication methods. Naapoy co-evolution of archaea and their viruses na na-observe. So example sa methanogenic archaea sa order Methanococales, the cellular mini-chromosome maintenance or the MCM helicase has apparently undergone accelerated evolution due to acquisition by a virus accelerated evolution as a viral gene, and reintegration into the host archaea, replacing the original MCM gene. In other cases, replication proteins encoded by archaeal viruses appear to share common ancestry with their archaeal counterparts, but the exact mechanisms of these are underexplored. So, the family B polymerase of pol B3 of halophilic and methanogenic archaea appears to have been recruited from archaeal codoviruses. For evolutionary considerations, studies on archaeal viruses allow insight into virus evolution, conservation of a characteristic virion morphology, and sequence similarity of some of the genes indicates that euryarchaeal viruses could share ancestry with tailed bacteriophages. Also, certain characteristics of the genomes as well as virus-host relationships of Crenarchaeal fossiloviruses indicate a common ancestry with lambdoid bacteriophages. Now let's go to the importance of archaeal viruses. Archaeal viruses are predicted to play a greater role deeper in the ocean and in the subsurface, where virus to prokaryote ratios and the quantity of virus-related DNA sequences in metagenomes are greater. There is evidence of high viral-induced mortality, mainly of Thomarchaeota in deep-sea ecosystems, resulting in about zero point 0.3 to zero point 0.5 gigatons of carbon release globally each year. The death of these archaea releases cellular content, thereby enhancing organic matter mineralization and respiration of uninfected heterotrophs. So in turn, this stimulates nitrogen regeneration processes, supplying 30 to 60% of the ammonia required to sustain archaeal chemoautotrophic carbon production in deep-sea sediments. Archaea and bacteria inhabit deep-sea sediments in roughly equal numbers, but virus-mediated lysis of archaea occurs at a greater proportion than of bacteria. So archaeal viruses may therefore be a major driver of biogeochemical cycling in the oceans. Immune system interactions. Nearly all archaea possess multiple immune defense systems. CRISPR-Cas in particular is near ubiquitous, especially in hyperthermophiles, as about 90% of sequenced archaea possess at least one CRISPR-Cas locus in their genome. These loci contain a leader sequence, alternating short identical sequences, which is termed repeats, and variable regions called spacers, which are usually identical to sequences taken from foreign DNA. CRISPRs are transcribed to produce CRISPR RNA, which can neutralize foreign genetic elements via base complementarity. Immunity to viruses granted from CRISPR-Cas systems is passed down to daughter cells as a form of inherited immunity. Archaeal viruses can, at least temporarily, avoid the CRISPR-Cas targeting system through changes in the target sequence. Some archaeal viruses carry CRISPR arrays, which likely prevent co-infection of the same cell by other viruses. Archaeal viruses also encode many proteins that modulate specific stages of virus-host interaction, including proteins that inactivate host defense mechanisms like CRISPR-Cas. Now we are on our last K topic, which is the potential pathogenicity of archaeal viruses. Now that we are reaching the end of our report, you may wonder, might archaea be capable of causing disease? Current data suggests that archaea are able to colonize and survive in humans. However, no concerted efforts have been undertaken to implicate archaea in human disease. Strategies can vary enormously, but they often define a signature for a family of pathogens. So, if some archaea behave as pathogens, they probably follow this basic scheme. So, first is the access and colonization. The ubiquity of archaea in nature provides them ample opportunity for access to and colonization of susceptible hosts. Methanogens, for example, have been identified among the human colonic, subgingival, and vaginal flora. Given the requirement for strictly anaerobic microenvironments, methanogens are likely to exist in human anatomic sites where well-characterized anaerobic bacteria flourish, possibly as co-inhabitants. Unique flagella have been identified for many genera of Archaea, and such flagella have more structural similarities to the type 4 pili of bacteria than they do to bacterial flagella. Multiple archaeal genomes contain partial TAD loci, which may encode TAD-like proteins involved in fibril formation and surface adherence. The second strategy is recognition by host defenses. Pathology due to microbial infection usually results either from direct damage to the host by the microorganism or from a host inflammatory response. The latter arises from a variety of interrelated mechanisms, including innate recognition of pathogen-associated molecular patterns, active induction of host response pathways by pathogen virulence factors, and acquired capabilities for antigen recognition. The first of these mechanisms may be the most primitive and highly conserved across a wide range of non-microbial virulence scenarios. Lipopolysaccharide has not been demonstrated in archaea, but when incorporated into liposomes that will form into archaeosomes, the unique polar lipids of archaea have been shown to serve as potent immune adjuvants both in vitro and in vivo. Such archaeal lipid structures demonstrate more potent adjuvant activity than conventional adjuvants such as liposomes or alum in vivo, regardless of the source archaeal species. For example, vaccination of mice with archaeosome-entrapped listeriolysin leads to rapid and prolonged specific immunity against Listeria monocytogenes infection and is superior to several other antigen delivery vaccine strategies. Next is the evasion of host defenses. Pathogens often rely on specific mechanisms to evade the host's immune response. Such mechanisms include the elaboration of toxins or enzymes, the use of antiphagocytic capsules, modification of the microbial outer surface, and induced or deliberate internalization into host cells. However, it is unknown whether archaea have specific means to evade human immune responses. Lastly, is the genetic potential of archaea as pathogens. So the virulence genes are often associated with transmissible genetic elements. It is plausible that archaea, in the context of a complex population of human bacterial flora, may have the potential for virulence gene acquisition by such mechanisms. Lateral genetic transfers between archaea and bacteria over evolutionary time periods have already been proposed. However, evidence of rapid real-time exchange of genetic material among or with the archaea is not available. So in this part, we're going to answer some major questions that are frequently asked about archaeal viruses. So the first is, are archaeal viruses different from viruses infecting eukaryotes and bacteria? The answer to this question is both yes and no. Known archaeal viruses can be roughly divided into two categories, those that are morphologically and genetically unique to archaea, and those with clear structural and genetic homologs to both bacteriophage and eukaryotic viruses. For example, many archaeal viruses infecting halophilic archaea resemble the tailed viruses belonging to the Codovirales order of bacterial viruses. Archaea-specific viruses represent some of the most unique viruses known, from bottle-shaped viruses to spindle-shaped viruses, and to two-tailed viruses whose tails grow once they are released from infected cells. Next is what diseases are associated with archaea and their viruses. Surprisingly, no archaea is known to cause human disease, and therefore no archaeal viruses are known to be involved with modulating pathogenesis in humans. Unlike bacterial pathogens, in which it is known that bacteriophage can contribute to pathogenesis, for example in the case of the cholera toxin, no such cases are known for archaeal viruses. None are known to cause disease because possibly it is that it reflects a lack of direct studies that investigate the role of archaea and their viruses in disease. The lack of clear evidence for pathogenicity of archaea and associated archaeal viruses in humans is in itself an intriguing observation which suggests a fundamentally different set of associations between humans, archaea, and their viruses. Next is, what role do archaeal viruses play in ecosystems? Determining the range of ecological roles viruses play in natural environments is a central question driving studies in environmental virology today. Acidic hot springs found in Yellowstone National Park provide ideal environments for such studies because they consist of simplified microbial communities made up principally of archaeal hosts and their viruses, and this allows for archaeal host virus interactions to be studied in their natural setting in the absence of other microbial players. Lastly, is the evolutionary history of archaeal viruses different from that of other viruses? In general, deciphering the evolutional history of viruses is a difficult task due to the lack of universally shared viral genes and the presence of extensive horizontal gene transfer among viruses and between viruses and their hosts. It is particularly difficult with archaeal viruses because we only have limited knowledge on archaeal virus genomes. However, there appear to be two groupings of archaeal viruses with distinct evolutionary trajectories. First, those viruses that structurally resemble viruses that infect members of eukarya and bacteria, and a second grouping whose viruses can only affect archaeal species. In order to gain a more in-depth understanding of archaeal virus evolution, we need to greatly expand our knowledge of viruses beyond the limited archaeal phyla that have been investigated to date.
And I guess that ends our presentation. Thank you for listening and have a good day.